Hello guys, welcome to the first video of NCERT MED Easy. Today our topic is gonna be electron transport system, also known as oxidative phosphorylation. First of all, we are gonna discuss what is ETS or what does it mean by oxidative phosphorylation. Keep in mind, both mean the same. The examiner will often ask the other term and expect you to know what does it mean. It will confuse you, so keep this point in mind. Now starting, what does our NCERT says? NCERT says ETS is a process where NADH and FADH are oxidized to give out energy and O2 is reduced by accepting proton. The metabolic pathway through which the electron passes from one carrier to another is called electron transport system. One important point over here is oxidation means losing of electrons and reduction means gaining of electrons. Keep this point in mind. This will be very handy in the later half of this video. I'll keep repeating this. On this point, the whole topic of ETS is gonna be very easy for you. Now, moving on. One question over here from the first para, which was a NEET PYQ. Where is the ETS located, or where are the ETS enzymes located in the mitochondria? The answer will be the inner mitochondrial membrane. Very important PYQ, asked twice, once in NEET 2016 and once in AIPMT 2011. Now let's come to the cycle. As you can see, ET's electron transport chain enzymes are present on inner mitochondrial membrane. One more PYQ over here is, where does the proton accumulation take place in the mitochondria? The answer will be the intermembrane space. All the protons are accumulated here from the matrix. This proton accumulation causes a charge development on the intermembrane space and a negative charge in the matrix. This is also called electrochemical gradient. As you can see, the NCERT does not mention it anywhere, but it's given in the diagram showing the positive sign and the negative sign. Now for the mechanism part, before that, the memorization part. What you have to memorize in this? Basically, everything you have to memorize is just about the complex names. Complex 1, NADH dehydrogenase, NADH to NAD plus. It is removing a hydrogen, it is getting oxidized, hence the name NADH dehydrogenase. Complex 2, succinate dehydrogenase. You remember in the citric cycle, succinate is dehydrogenated and it releases FADH, from which FAD comes, and that is the complex 2. That is complex 2. The complex 3 is complex B, cytochrome BC1. You have to remember this. And the complex 4 being cytochrome C oxidase. Cytochrome C is a protein, small protein attached to the outer surface of the inner membrane. Outer surface of the inner membrane, and it acts as a mobile carrier for the complex 3 to 4. It transfers the electron from complex 3 to 4. The cytochrome C complex takes the electron. Remember, taking electron means it gets reduced from the complex 3. Then it accepts the electron, it travels to complex 4, it gives the complex 4 its electron, hence getting oxidized. Hence complex 4 is named as cytochrome C oxidase. Remember guys, complex 4 is also known as 2 copper centers. This was a PYQ, very important PYQ. The mock test will be based around this question, I can guarantee that. Now coming from the starting, complex 1. We know that NADH takes 380, produces 3 ATP. So when electron is given from the complex 1, it travels through ubiquinone. Ubiquinone is basically a carrier protein. It carries electron. When ubiquinone receives an electron, means it gets reduced. It becomes ubiquinol. This is a possible question. They can ask this. Remember the name. Reduced ubiquinone is called ubiquinol. Now ubiquinone also accepts the electron from complex 2, which also converts into ubiquinol. Now the ubiquinol gives out electron from the cytochrome BC1 complex to cytochrome C, reducing it. Then cytochrome C transports its electron to complex 4, oxidizing itself. From there, the hydrogen protein goes from F0 protein to F1, which is complex 5. Complex 5 is nothing but ATP synthase. F0 and F1 are, we'll see in the next page. F1 is peripheral, F0 is in the inner mitochondrial membrane, embedded in the inner mitochondrial membrane. F1 has complexes, it couples the hydrogen. So if two hydrogen are coming from here, it couples it makes it 4 hydrogen. And remember guys, 4 hydrogen gives 1 ATP. Hence the diagram over here and over here is clearly justified. It's not a mistake. This is a big confusion between students that this is a mistake in NCERT, but this is not, because F1 couples the number of hydrogen, making it 4 hydrogen. From there, ATP is synthesized from the ADP and the phosphorus. Now coming to the extra points, this was the whole mechanism. That's it. That's the ETS. I'll give a PDF for this NCERT highlighted. Make the pink highlights are all the PYQs. Make sure to go through them. Write it, mark it in your NCERT, write it in your notes somewhere, and this should be enough for the NEET exam. They won't ask outside of this. It's not necessary to go very deep into this process. This is a very complex process. So just do as much as it's there in the NCERT. The extra points being, this whole process is an aerobic process, but the role of oxygen is limited to the terminal stage of the process. The oxygen is not used anywhere except the last stage, where it acts as a final hydrogen acceptor. This makes oxygen very, very important, but the role is very, very minimum. These are both the PYQs I marked. One confusing line in NCERT over here, I'll explain. Like light phosphorylation, what happens was light energy is used to convert ADP to ATP, and hence energy was produced. Over here in respiration, the oxidation and the reduction of the complexes are used to create an electrochemical gradient from which the energy is taken out. Hence the ETS is also called oxidative phosphorylation. Very, very important. They can ask this as assertion reason. This line, production of proton gradient required phosphorylation. In respiration, it is the energy of oxidation reduction utilized for the same process. It is for this reason that the process is called oxidative phosphorylation. That sums up your ETS. I hope you like the lecture. If there are any doubts, you can comment, and if there's a major doubt, I'll make another video about it, or I'll try to solve it in the comment section.